abstract session. We are pleased to announce that the call for abstracts was a success. We received 31 submissions related to eosinophil biology, gastrointestinal eosinophilic disease, and other hypereosinophilic conditions. Our scientific committee has scored the abstracts and selected the top-rated ones for live oral presentations. Next slide, please. The first 3 presentations will be 10 minutes long with 5 minutes for discussion. And then there will be 3 additional 3-minute presentations. And although these will be live, they will be discussed during the breakout sessions that will follow immediately the main session. All of the remaining abstracts will be presented in a 1-minute pre-recorded format in the breakout rooms. At the end of the live presentations, we will display the names of the 3 breakout rooms and the abstract titles of the presentations that will be presented and or discussed in each room so that you can decide where you want to go. There will be a moderator in each room to help with the flow of the discussions, but you will be able to interact verbally if you unmute yourselves, and you can also show your faces. At the end of the half-hour breakout session, we will all reconvene on the main platform for final announcements, including the distribution of awards for the top-rated presentations. Please take a rapid look at the housekeeping rules that are found on the next slide. The main messages are to please stay muted throughout the main presentations, and don't hesitate to use the chat box for questions or comments on these main presentations, as there will be a moderator who will actually voice these questions and comments for you. It is now my pleasure to introduce the moderators of today's main session, Panis Khoury and Manali Mukherjee, who have both been instrumental in making this abstract session happen. Panis Khoury is Program Director and Associate Research Physician at the National Institute of Health. Manali Mukherjee is Assistant Professor of Medicine at McMaster University and Affiliate Scientist, both working, of course, on eosinophil biology and eosinophilic disorders. With no further ado, I will leave you in their very capable hands.
Thank you, Florence. So for our first presentation, it's my pleasure to introduce Dr. Tetsuo Shoda from the Cincinnati Children's Hospital Medical Center in the United States. He's going to present on a multi-site study of colon molecular profiles that establishes eosinophilic colitis as a unique disease entity. Thank you, Tetsuo. Please go ahead.
Oh, sorry. Let me share my screen. Can you see my screen?
We can. Go ahead.
Okay. So let me start. Thank you, Chairperson. Hello, everyone. I'm Tetsuo Shoda from Rosenberg QR Lab in Cincinnati Children's Hospital. Thank you for giving me this opportunity to present our study here. Today, I'd like to talk about the colon molecular profile in eosinophilic colitis. Let me start with a brief background about the study. As you may know, eosinophilic gastrointestinal diseases, I'll say EGID today. EGIDs are clinical pathologically characterized by massive eosinophilic infiltration into the GI tract. According to the site of infiltration, EGIDs are classified into eosinophilic esophagitis, gastritis, duodenitis, and colitis. Today, we focus on eosinophilic colitis. I call it EOC in this presentation. The disease pathogenesis of EOC remains poorly understood in spite of a high disease burden. The diagnosis of EOC is not straightforward. Except for the esophagus, eosinophils are present in GI mucosal biopsies. Eosinophils are the highest in the ascending colon, and it tapers to lower levels in the left sigmoid colon. And it has been proposed that the diagnostic criteria for EOC include at least twice the normal peak eosinophils. But unlike the EOE, there are no agreed upon diagnostic criteria at present. I'm not sure. Okay, yeah. So we thought further understanding of the EOC pathogenesis would be needed. In terms of the EGID research, substantial progress has been made using gene expression profile of tissue biopsies from patients. EOE transcriptome has contributed to understanding of disease pathogenesis. For instance, EOE transcriptome can distinguish EOE, monitor the disease activity, and N-type EOE. Following the progress of the EOE transcriptome, the EOG gastric transcriptome was also identified, and it has been utilized for diagnosing and monitoring EOG. So we thought more study to understand molecular profile in EOC would be helpful in the field. In this context, we aim to establish the EOC transcriptome, their functional enrichment, and its potential utility for molecular diagnosis by answering following fundamental questions for EOC. This is below. To approach the aim, we analyzed the colon sample from subjects with EOC and controls, including normal and Crohn's disease. From multiple institutes associated with figure, 87 colon biopsies from 61 subjects were included in the study. Based on the RNA sequencing data and histologic data, we tried to establish EOC transcriptome and assess its clinical significance. The first question is if EOC is a distinct disease entity or not. To obtain key molecular profile for EOC, we generated an RNA sequencing data from active EOC and controls. Then we identified about 1,000 genes that were differentially expressed compared to controls. And also unsupervised clustering analysis showed clear separation between the active EOC and controls, as shown here. And subsequently, to compare with the EOC transcriptome, we also identified a transcriptome for Crohn's disease using the same approach as we took for EOC. As shown in the Venn diagram, EOC transcriptome showed distinct from CD transcriptome, and also PCA plot and clustering analysis showed the robust separation of active EOC, Crohn's disease, and controls. Taken together, these data suggest that EOC as a unique GI disease. The next question is the molecular relationship between EOC and other EG. Also, we wanted to know if EOC shows the evidence of allergic type 2 immunity. So to determine the relationships, we compared the EOC colonic transcriptome with the previously published EOE esophageal transcriptome and also EOG gastric transcriptome. Notably, there are very minimum overlap among transcriptomes of these three EGs, EOE, EOG, and EOC. Common genes were only 9 genes. It's 1% of EOC transcriptome. To make sure if EOC is characterized by type 2 immune profile, we first focused on eosinophil and type 2 related gene expressions, as shown in this heat map. But type 2 related gene expressions were not dysregulated in EOC. So we didn't find evidence of type 2 immunity in the EOC transcriptome. Next, we hypothesized that the specific element of the EOC transcriptome will be correlated with disease function, since we didn't find the type 2 immunity. To identify the molecular pathways associated with EOC, we performed functional annotation enrichment analysis. For downregulated genes in EOC transcriptome, the highest enrichment were decreased cell cycle pathways. And for upregulated gene in EOC transcriptome, we found the increased apoptosis pathways. The finding of reduced cell proliferation was unexpected to us because other EGID and IBD usually show increased cell proliferation. So we tried to make sure of this finding by staining of patient biopsy using the Ki67 it's a marker of cell proliferation, and cleaved caspase 3, it's a marker of cell apoptosis. As shown here, compared to controls, EOC showed reduced cells with positive Ki67, and in contrast, cleaved caspase 3 positive cells was significantly increased in EOC compared to controls. Taken together, we found decreased cell proliferation and increased apoptosis in EOC biopsies. It's consistent with pathway analysis. The final question was if EOC transcriptome is clinically useful. To generate quantitative value to reflect molecular changes, we developed the EOC score on the basis of the EOC transcriptome. The EOC score was specifically increased in the patients with active EOC compared to inactive EOC and controls. It's indicating its reversibility with disease remission. To further assess its clinical utility, we compared active EOC and the subset of Crohn's disease having higher eosinophil levels in the column. Because Crohn's disease having high eosinophil levels is clinically challenging to differentiate from EOC. Although there was no difference in the peak colonic eosinophil count, the EOC score clearly separated these two groups, consistent with unsupervised clustering analysis, as shown in this heat map. And taken together, these data suggest EOC transcriptome as a function of disease activity and differential diagnosis. So in summary, we identified EOC transcriptome and established EOC as a unique GI disease. Mechanistically, we uncovered the EOC is not related to type 2 immunity, but rather apoptosis and reduced cell proliferation. And we also showed the potential utility of EOC transcriptome for disease monitoring and diagnosis. And here is acknowledgement about this study. And that's all I have to say. Thank you for your attention.
Thank you so much for that beautiful talk. We're going to take some questions and answers. And I already see that there's something in the chat already. And so this is from Dr. Cook Mills. She says, although only few genes are in overlap, are these genes ones that can drive all three types of eosinophilia? But the other non-overlapping genes are because tissues respond a bit differently to the drivers.
So the question.
So I think what she's asking is, are there some genes that are not overlapping in the EOC transcriptome, but may still be driving a similar process? For example, the fact that you see eosinophilia and EC.
The overlapping gene, for example, the CLC is a kind of eosinophil specific gene. Is of course it's a common gene. So of course eosinophilia these common genes are associated with common pathways to drive eosinophilia. But some and also basic kind of inflammatory signaling also shared among these diseases. But we didn't find about especially type 2 immunity. Unexpectedly, we didn't find the evidence based on this analysis.
Okay, thank you. I think there's another question that kind of goes along the same lines by Dr. Alam, who says, can you comment on the genes that drive eosinophilia in EOC, since type 2 genes are not involved? So I think he's referring to the fact that the IL-4, 13, and were not elevated, but you still see CLC and CCL11 in your heat maps. Can you comment on that?
Yeah, that's a nice question, but at least for now, I don't have answer for what is what driving the eosinophil exactly. Yeah, this is for further research. Actually, but CCL11 is upregulated, so probably CCL11 is one of the main chemoattractant for for eosinophil in this disease.
That's great. So one question from Dr. Seves says, can you comment on the disease activity of the EOC cohort and if you think this might affect the eosinophil trafficking signals? I think the EOC is the gene set. I'm not sure, or maybe she's talking about the eosinophil colitis cohort. If you think that this may affect the eosinophil trafficking signals.
Cohort.
Sorry, Tetsuo, what I wanted to ask you is if you think the activity state of the biopsy, so like were they all active EOC biopsies, and if they're not, do you think that was affecting what you saw in terms of the trafficking signals?
I see. So we used only at least the more than twice eosinophil counts as compared to the upper limit of normal. So usually their eosinophil level is more like more than 100. So it should be. I thought it should be activated, active disease state, and
so
also and also we compared the active and inactive. The transcriptome, show the reversibility. So I think it's activated group.
I think there are just two more questions that we might have time for. One very interesting question asks if there are differences like sex or age that can explain the different transcriptomes in EOC and other Crohn's disease cohorts.
So far our cohort is not, many sample. There are not enough sample to explain these differences. So because it's very rare disease, so we need to collect more sample to explain these differences.
Understood. And then the final question that we have time for, Dr. Rufo says, very nice talk. Do you have an explanation for why Siglec does not appear among upregulated genes in this particular form of eosinophilic GI disease compared to others?
Siglec, I think Siglec 8 was slightly up, increased compared to control, but it's not, there are some variability in the biopsy. That's why it's not, in the list.
Thank you very much. I think that's all we have time for before we go to the next talk.
Thank you so much.
So we had a good start already. Our next presenter of the top abstract is again from Cincinnati Children's Hospital Medical Center in the USA. It's Dr. Natalie Ben-Baruch Morgenstern, who will be speaking to us about single-cell RNA sequencing of mast cells in eosinophilic esophagitis, revealing local proliferation and activation that persists in remission. So over to you, Natalie. Looking forward.
Thank you so much. Just a second, I'm going to share my screen. Can you see it?
Yes.
Oh, maybe I should share my presentation instead. Just a second. Can you see it?
Yes, we can. Thank you.
Perfect. So first of all, I want to thank so much for this opportunity to present my project. A short background. So eosinophilic esophagitis, I'm just going to put it over here. Eosinophilic esophagitis is inflammatory disease that's triggered by food antigen. It has a T2 profile and unique histopathology that includes accumulation of eosinophils, expansion of mast cells, and additional pathologic and tissue remodeling in the disease. Currently, there is no cure, and the treatment promotes the remission of the disease. However, when treatment regimens discontinue, the disease recurs. So we were interested to expand our knowledge about the expansion of mast cells, because until now we know that it presents an expansion in the tissue, but many of the characteristics are missing. So our aims was to characterize esophageal mast cell genetic heterogeneity, assess mast cell composition across the spectrum of EoE activity, and evaluate the potential of mast cell contribution to the tissue. So our approach included collect biopsies from esophageal, from control patient active EoE in remission, and we performed comprehensive analysis of flow cytometry, immunofluorescence, and single-cell RNA sequencing in order to answer our questions. The first thing that we looked is single-cell RNA sequencing, and we were able to identify at least 7 populations in the tissue. One of them was the mast cells, and we were interested to compare the transcriptome of mast cells to the other population and to see which inflammatory proteins are enriched due to the increase of mast cells in the tissue. So in the left, you can see the UMAP of the transcriptome of the single-cell RNA sequencing, and in the right, you can see the heat map that we compare between mast cells and the other the main cell population that we identify. And we saw that in active EoE, each line is a different patient. In active EoE, we see enrichment of cytokine and growth factor, proteases, histamine and lipid mediators and receptors. And for our surprise, we also see that IL-13 is extremely enriched in mast cell. It's significantly more enriched in mast cell compared to the other population in the tissue. While until now, we knew that IL-13 likely generate mainly by the T cell, here we can see that mast cells are also important contributor to the increase in IL-13 in the transcriptome level. Next, we wanted to assess the expansion of the mast cells in all 3 states. So we performed flow cytometry, and we found the mast cells by the markers of Fc epsilon 1 and c-KIT. They are both high to these markers. And we saw that the expansion of mast cell is not only occurring in active EoE. There is significant increase in active EoE, but this increase is actually persistent remission. We looked on the by immunofluorescence also in the tissue. We stained the tissue to tryptase as a marker of mast cells in magenta, and DAPI is the nuclear staining. And we saw that similar to what Dr. Aceves and Dr. Abonia demonstrated back in 2010, in the lamina propria, mast cells exist also in control, and there is not a significant difference in the levels of mast cells in the lamina propria. However, in contrast, in the intraepithelial compartment, mast cells are increased in active EoE, and we definitely see the trend also of increased level in remission in the intraepithelial compartment. So we were interested to focus on the mast cells. So we depicted only the mast cells from our RNA sequencing dataset, from the single-cell RNA sequencing dataset, and we wanted to see if we can explain this expansion, where it's coming from. And we saw the first thing that is notable is this population here in purple. When we looked on the transcript, we see that it's enriched with proliferative genes, including MKI67, TOP2A, and etc. It means that that muscle actually, mature muscle actually can proliferate in the tissue. We wanted to see if this exists also in the protein level, and not only on the transcriptome level. And we saw that when we stain to Ki67 and to tryptase here in magenta and the Ki67 in green, we see that we can identify this proliferated mast cell also in the tissue, and they are enriched in active EoE, and they're still enriched. They are still increased in remission, but significantly lower in remission compared to active EoE. Next, we wanted to continue to evaluate the heterogeneity of mast cells in the tissue. We looked on all 4 populations, and we named them according to their appearance in the states of the disease. We found that the resident mast cells, we named it resident because they appear in all three states, in control, active EOE, in remission. In active EOE, we found two additional subpopulation of mast cells, including the transient and the persistent, and also the proliferative. And we named the transient because it's abolished in the remission, and the persistent because it still continued to persist in the remission. We also found, I'm not showing it here, but we also found that the proliferative mast cells are not part of, it's not independent population. It's likely to be part of the transient and in less extent also from the persistent, but it's not a unique cluster. And we wanted to look on the markers of this population, and we looked on the markers of each population in the different states, and we found that there is increased heterogeneity in EOE. They are enriched in different markers. For example, the resident enriched with tryptase, while the persistent in both states is enriched with cathepsin G and CMA1. Of course, there is some variability between states, but we can definitely say that it's completely different population, and there is increased heterogeneity. So we concluded that esophageal mast cells are composed of at least 3 subpopulations, 2 are EOE specific. So it was really obvious that there is enrichment in inflammatory genes during active disease and remission, and we wanted, as you can see here, in active disease, there is enrichment of many proteases and other inflammatory. In remission, enrichment although indifferent, but there's still enrichment compared to the control. And we wanted to look on that and to get approval to our to what we saw. So we used activation signature that was published in Immunity in 2019, that they took progenitor from the peripheral blood and differentiate them to mast cells and demonstrate the activation signature. We took this activation signature and applied it on our mast cells from the single cell RNA sequencing data. And we saw that indeed mast cells in active EOE are demonstrate increased activation signature compared to control, and this activation signature is persistent in remission. It's still more activated compared to control. So if we have mast cells in the remission, when we have mast cells in remission, they are more activated compared to control. They persist to be activated. Next, I'm going back for a second to the first slide that I show you, that mast cells are enriched with IL-13 in the transcriptome level, and we wanted to see what we see also in the protein level. So we looked on the tissue by immunofluorescent. We stain IL-13 in green, tryptase in magenta. The combination create white, and we saw, for our big surprise, that in the lamina propria from control and active EOE, we see the mast cells, the same levels, but they were loaded with IL-13. So the mast cells, all mast cells were loaded with IL-13, and all the mast cells, almost 100% of them were IL-13 positive. This is in contrast to the epithelium, that when mast cells were present in the epithelium in control, not a lot of them were present, but when they were present, we saw IL-13 in the tissue, as you can see here in the percentage. In the active disease, we saw much more mast cells. That's why we saw more IL-13 positive mast cells. However, when we looked on the percentage, we saw that the majority of the mast cells in the intraepithelial compartment were lacking IL-13. So we concluded that mast cells likely secrete the IL-13 because they have the ability to secrete the IL-13, so they likely secrete the IL-13 in the epithelium during active EOE. So our summary and conclusion is that in homeostasis, we have the resident mast cells localized mainly in the lamina propria. They demonstrate quiescence profile. During active EOE, we see at least 2 other populations that are expanding in the tissue, the transient and the persistent, both of them linked to proliferation. And we see increased activation of all three populations in the tissue. This increased activation is persistent remission, and the persistent population is still present in the remission state. Both of them are increased activation. Thus, our hypothesis is that that they are likely poised to be activated upon treatment withdrawal. So if we conclude, our main take-home messages are: esophageal muscles are dynamic population that can increase their numbers, heterogeneity, activation, and compartmentalization during EOE. Esophageal muscle undergo in situ proliferation. In remission, muscle persist in pro-inflammatory states, poised to reinitiate inflammation in EOE. Thus, they may be essential in disease relapse upon treatment withdrawal. Esophageal muscle constitute a prominent source of inflammatory mediator, including IL-13. Assessing muscle level and restraining their activity should be considered for treatment of EOE. I want to great thanks to my mentor and my PI, Dr. Rothenberg, and to our collaborator from Harvard University, Nora Barrett and Dr. Daniel Doyer, and to the other lab members that participate in this project. And for you to listen.
Thank you, Natalie. That was a very, very interesting talk. And we have a number of questions popping up in the chat box while you were speaking. The very first came from Dr. Ackerman, and I kind of have the same burning question for you. Can you tell us why there was no eosinophil signal in your single-cell RNA transcriptome?
So this is, we use the 10 genomic, the version 2, and this specific, the 10 genomic have hard time to detect eosinophils. There are few possibilities. Multiple papers demonstrated that by the 10 genomic, it's really hard to detect eosinophils. Many failed, including our collaborator from Harvard University. And it's likely due to the high elevated levels of RNases in the tissue, or even the lack of high amount of enough of RNA to detect. However, there are additional 10 genomic, additional single cell RNA sequencing methods that demonstrate successful success in eosinophils, to detect eosinophils, including the recent paper from Science Immunology that used the 10x. They claim that the method is more sensitive and it's more and treat to the eosinophils more gentle using the sequel compared to the droplet methods. So currently we don't see it with the 10x. We might be able to do that with the sequel or other methods of single cell.
Yes, because given the name as eosinophilic esophagitis and there has been eosinophil seen in biopsies,
they are there. I'm demonstrating, not in this presentation, but in every single biopsy that were used to this research, we saw eosinophils by other means, including histology or flow cytometry. They are present there. We validated in each and every active disease that they are present. Just the 10x cannot detect them. Also, the 10x, there is some limitation to the 10x, and each version has its own limitation, and that's something that need to take into account when you do single cell RNA sequencing. You have to know the limitation and to try to compensate them by other means.
I see. Thank you. I think Gunnar Nilsson was also asking you about the same regarding which method was used. I think he got his answer. And I guess even Tenenbaum Kim was also asking the same question. Dr. Bruce Bauchner has been asking you whether you found any cell-specific Siglec signals, Siglec 6 or 8, and how long were subjects in remission at the time of the remission biopsy? I think this is the last question I'm going to ask in the interest of time.
So Siglec, in the single cell RNA sequencing, we were not able to detect eosinophils. I searched for Siglec 8. The muscles can, I think that they can detect it, but not specific other than the other population that express it. Eosinophils are not there. We tried in adding methods even to lower the detection, that the transcript, just to I don't remember cyclic 8, cyclic 6. I need to examine that. I need to check. I looked on, there are so many genes there. I think that I looked, but I don't remember. I'm sorry. I can check after.
Dr. Bruckner agrees with you that it's extremely difficult to find eosinophils on the single cell RNA-seq, even nasal polyps they fail to see. So I'm going to go and hand it over to my co-moderator Panis for the next talk.
Thank you so much. I appreciate it.
Thank you. So for the next talk, it's my pleasure to introduce Dr. Katrin Meyer-Barber from the NIH in Bethesda, Maryland, United States. She's going to speak on eosinophils as an integral component of the pulmonary granulocyte response in tuberculosis and promote host resistance in mice. Take it away, Kat.
Hello, thank you, and I'm sharing now. And there you go. Thanks so much for selecting my abstract and allowing me to share my research. And I'm particularly interested in getting your guys' expert feedback on this project. So just to remind everyone, I know that's not a disease you typically think of when you study eosinophils. TB is one of the leading causes of mortality worldwide due to a single pathogen. And I don't think actually COVID has changed that. If anything, COVID really has exacerbated this problem because just like COVID, TB is spread by aerosol, and there have been a lot of resources diverted from case recognition. So despite being around for a long time, and of course affecting not necessarily the same affluent, economically affluent areas as COVID, it's been really quite difficult to manage the disease. And we have drug resistance emerging, of course, but then you're also, you know, have the HIV co-pandemic, which really highlighted that we require CD4 T cells for optimal immune protection, and that this is a predominant Th1 immune response. We know gamma and IL-12 is super critical. My lab is really interested in innate immunity, and I want to just highlight that, of course, myeloid cells are super important because they're the ones that are actually infected and have to eliminate the bacteria. So receiving instructions and the communication amongst cells is super critical. And so my lab is primarily interested in understanding the innate inflammatory events that are driving pathology and the early events after pulmonary exposure, but also which innate immune subsets are involved at each step, and which cytokine and lipid mediator pathways are playing a role. I'm not going to talk about any of the IL-1 type 1 or prostaglandin pathways we've been studying, but I want to really highlight some of the innate immune cell types we came across, and one of them is the eosinophil. So we're really trying to understand which innate immune subsets control host protective versus detrimental inflammation, and how we can target this ultimately for adjuvant design or host-directed therapies. And one of the long-standing interests was, you know, using mouse models, we have a canny ability to really dissect by multidimensional flow cytometry the different innate immune players. But we've been always struggling with trying to really recapitulate the human pathology. And so one of my long-standing goals was to set up a collaboration where I can actually study the innate immune cells directly in human lung tissue. And so to this end, you know, I found a wonderful place where I was able to do just that in the Shanghai Public Health Clinical Center in Shanghai. This is a multi-building campus where you have the surgery happening in one building, and then we can take the resected human lung tissue directly to the next building where there is a state-of-the-art BL3 laboratory where we can do fresh cell isolation and trying to understand what's going on. So this project has been really driven by two outstanding biologists in the lab, Andrea Bora and Edel Castro. And so just, I mean, you guys don't need this background in detail, but just to, you know, when you look in circulation, we can distinguish eosinophils from neutrophils based on cyclic 8 expression. And we know that neutrophils really outnumber eosinophils in circulation. We were surprised to see, however, when we looked at TB resected lung tissue, that in some of the areas that we were able to identify based on macropathology, that the eosinophils were actually outnumbering neutrophils at some point. And this is an example of a granuloma where you can clearly see we have many more eosinophils than neutrophils. And that really got me curious, like what do we really know about eosinophils in TB, which is experimentally speaking, very little. So we started out by looking in a multiple clinical cohorts. I'm just highlighting this year one in Cape Town, where I'm showing you the ratio, but I want you to just know the actual number of eosinophils mirror this. So we found that when you have active TB disease, it means you have a TB antigen-specific test and you can detect TB in the sputum. We actually get a decrease in eosinophils, and this is of course exacerbated by showing you this ratio of neutrophils. So we know that when you have active disease, the total number of eosinophils go down compared when you're latently infected. And so we were wondering, well, if we see them enriched in the lung, but they're going away in the blood, maybe they're going from the circulation to the site of infection. Because we had this unique opportunity to look in human lung tissue, we had a few patients where we could simultaneously assess by flow cytometry the frequency of eosinophils in neutrophils in tissue versus circulation. And indeed, in basically all of the patients we looked at, eosinophils were selectively enriched in TB-infected lungs. So then we wanted to expand this to other models and wanted to focus on models that are known to form bona fide granulomatoous structures. As you know, this requires tissue remodeling. Some of these granulomas have fibrotic cuffs. So maybe this is a connection as to why we might see eosinophils here. And so we confirmed the flow cytometry with histology. Here I'm showing you EPX staining in human lesions from an independent cohort. We then started to really work in the non-human primate model, and we could also find eosinophils. And we were even able to identify eosinophil-like cells in the Marinan model in zebrafish. So to really quantify this though, in the non-human primate model, we had to develop a quantitative method, which is flow cytometry, of course. And as you know, human eosinophils express cyclic 8 at EPX, but cyclic 8 does not stay in non-human primate cells. So we devised and just utilized the EPX in the non-human primate model to really quantify now at the very detailed level the eosinophil response after MTB infection in the non-human primate model. So when we looked in the granulomas from MTB-infected macaques, one thing I want to highlight here is that there's a pretty huge diverse response, and we have granulomas where basically up to 10% of all the immune cells are eosinophils, and some granulomas where we have very few. So there's a massive diversity, and that's something that's been recognized in the TB field for a long time, that granulomas are highly diverse, and that's something we all would like to understand. And so when we look in the, if they're functionally activated by CD63 surface expression, which is a surrogate for degranulation, you can see here, of course, they do not degranulate in the blood, which is great. They do not, they degranulate a little bit more in the bowel fluid, but there's very little bacteria here compared to the granuloma, where we see a substantial amount of bacteria. So at the site of MTB bacterial load, we see eosinophils can get activated and degranulate. So this was really exciting. So maybe they actually see TB infection. And so we wondered if they are recruited to the infection directly because of TB, and it's not just a tissue remodeling response, what role do they play in host resistance? And so, of course, this is something a bit more complicated to address in humans or even non-human primates. So we really went back to the mouse model now. And here I just want to highlight that even in a B6 mouse model with low-dose aerosol, we see over time an accumulation of lung parenchymal, so truly lung-resident eosinophils after low-dose aerosol infection. And I want to just point out that overall, this is a highly neutrophilic system compared to even human and non-human primates. As you can see here, 4 weeks after MTB infection, the numerically eosinophils do not respond. This is not a typical, this is not a type 2 environment. The cells that are there, however, they really are being recruited into the parenchyma, here showing you the intravascular staining technique. And this just accelerates over time. And I also want to point out that the eosinophils are not actually infected directly, as has been shown with Staph aureus or other extracellular infections, and we can actually very sensitive, with a very sensitive method, identify infected cells. So we know they're not directly infected, but they are clearly accumulating over time. And so now that we have the ability to look in mice, we can use eosinophil deficient mice and ask, what do they really do? Are they protective, or are they maybe contributing to pathology? And so one of the things I want to highlight here, so with this is the low-dose B6 model. We used the delta double GATA mice on the B6 background. A normal time point that we study in the TB field is a 4-week time point. And I want to point out here that there is no difference in bacterial loads. And maybe this is one of the reasons why this has not been published previously, right? Because if you do a standard time point and you don't see a difference, maybe you abandon mission. Of course, because we started from humans, went to non-human primates, we knew we needed to be a bit more, nuanced about what's really going on. And long and behold, as you wait over time, as they are accumulating in the lung tissue, we actually can see that without eosinophils, there's an increase in bacterial loads. And that translates to significant decreased survival, whether it's on the Black 6 background or the BALB/c background. And we confirmed this with the Phil mouse model as well. And then the other thing I was concerned about, perhaps, as you can see, these are quite long time frames, even though I know without infection the mice live just as long. I just wanted to make sure this is infection related. So we used a high dose model. When we add even more MTV, the differences became even more pronounced, arguing that this is a true requirement of these eosinophils in response to MTB infection. So you can see we have a very clear difference in bacterial load around 2 to 3 months after infection, and the susceptibility of the mice, either Black 6, BALB/c, or Phil mice, is also quite accelerated still. So to just summarize, we think that eosinophils have not been studied systematically in a Th1 environment or intracellular bacterial infection, and we actually found that they were fairly abundant, or could have been, in certain TB pathologies from humans, quite abundant. We then went on to the non-human primate model, where we saw that they're also quite abundant in some granulomas, and we found evidence that they're functionally activated and can degranulate. And then we finally went to the mouse model, where when we remove eosinophils, we can show that they actually promote host resistance in the mouse model. So we actually don't know what the mechanism is, and we think there might be multiple mechanisms at play in the non-human primate, where you have granuloma structure, compared to mice, which actually do not form these structures. We've done some RNA-seq studies to look at the mechanism, and I can tell you it's not straightforward. We found neuronal associated pathways primarily activated, and of course, lipid mediators. That's something we're following up. So with that, I want to hopefully get some great feedback from you guys on the directions we could take this. Thank my small but mighty team, and want to highlight that we're actually recruiting. So we're looking for postdocs. So thank you, everyone.
Thank you so much. So we have a few minutes, so I'm going to ask some of the questions and hopefully get some brief answers so we can get to all of them. Are granuloma formation affected in eosinophil deficient mice? From Dr.
So mice do not form granulomas per se, and we've looked at the pathology, there was no difference. So this is something we would have to address in the non-human primate model.
Great. Are eosinophils playing a role in clearance or repair and at a later stage, or is the neutrophil exhaustion perhaps occurring in the tissue?
That's a good question. So we have not seen any differences in the neutrophils' ability to do anything in these late time points in eosinophils. We very carefully looked at the macrophages. We couldn't find a cell intrinsic role, whether they saw eosinophils or not at these late time points. So we actually don't know what the cellular mechanism is. We are wondering whether the eosinophils are interacting with some airway sensory neurons, and that they that there is a larger network at play of neuroimmune interactions that allow lung function, and somehow that's connected ultimately to control.
That's very interesting. Dr. Rothenberg asks about perhaps looking at the phenotype of the eosinophils that are accumulating. Have you done any work in that regard?
Yeah, so we we looked at primarily the tissue resident eosinophils, and those are the ones that I guess have been described to be more regulatory based on the fact that they've just entered the tissue. But we don't, we can't see any difference in whether we look at them in TB versus any other model. So there's nothing unique about the TB eosinophils that we can see at this point.
Okay, and then do you observe eosinophil products such as charcoal, Leiden crystal in the human granulomas of TB? I think we had a pause. I don't know if anyone else can hear me or if I'm paused, but I think we've reached time. So I'm going to hand it over to Dr. Mukherjee to take over for the next talk. And there are some additional things in the chat, so if you'd like to take a look at those and we can address them during the breakout session. Thank you very much.
Thank you, Penise. We are starting off our 3-minute sessions, so we will not be having immediate question-answer series right after that. So we will have 3 minutes at the end of all the 3-minute presentations, or we have to go back to our breakout room sessions to have those discussions and any burning questions around that time. So our first presenter for the 3-minute presentation is Dr. Juliette Tenenbaum from the National Reference Center for Hypereosinophilic Syndrome from France, and she's presenting on FIP1L1 PDGFR-associated Hypereosinophilic syndrome as a treatable cause of watershed infarction.
Hello, I am Juliette Tenenbaum. Today I will report to you our experience on ischemic strokes in the setting of FIP1L1 PDGFR alpha myeloid neoplasm with eosinophilia. The article is currently in press in the Stroke Journal. As you may know, FIP1 fusion transcript leads to the uncontrolled proliferation of the eosinophilic lineage, resulting in organ damage. We recently conducted a retrospective study of 151 patients with this transcript in France, and we observed that 10% of patients experienced an ischemic stroke. Strikingly, most of these strokes had a distinctive pattern on the brain MRI that was bilateral watershed distribution. We further reviewed the medical files of these patients, and we identified 16 males with a median age of 51 years old. Interestingly, the ischemic stroke was the initial manifestation of the myeloid neoplasm in 81% of patients, and none was under imatinib at the time of stroke. Clinical presentation was mild, with a median neurological score of 4, and patient had low to intermediate cardiovascular risk as assessed by score scale. When compared with the patients without stroke in the cohort, patients with stroke had significantly more frequent cardiac involvement, as 50% of the patients were diagnosed with eosinophilic cardiomyopathy, consisting of myocarditis or endomyocardial fibrosis, but only one patient had a documented left ventricular thrombus. 15 patients were treated with imatinib, and 100% achieved molecular remission over the median follow-up. Only 2 patients presented with ischemic stroke recurrences, but they had a different MRI pattern, and they occurred in patients with normal blood eosinophilic count and new cardiovascular risk factors for the ischemic strokes. Interestingly, in the literature, watershed infarcts have been reported in numerous eosinophil-associated diseases, such as acute schistosomiasis, ANCA-negative VGPA, lymphocytic variant HES, and DRESS, among examples. And we noted that in these reports, as well as in our case series, only roughly 50% of the patients had documented cardiac involvement. And it's interesting to note that this distribution, this pattern with watershed distribution is unusual for a cardioembolic event, which led us to hypothesize that a multifactorial process is involved in this watershed infarct, and it could involve microembolism and local vascular toxicity. As a conclusion, we would recommend prompt initiation of imatinib in the setting of FIP1 myeloid neoplasm, because it might prevent ischemic stroke recurrences. Thank you for your attention. I'm now open for questions.
So as mentioned, we're not going to take questions right now, but in the breakout rooms. So I'd like to introduce next Dr. Rafael Alam from National Jewish Health in Colorado, United States. He's going to talk about the molecular and epigenetic mechanisms and innate lymphoid cell memory for allergen-induced eosinophilic asthma.
Do you see my, I have shared, give me a second. Do you see my screen?
Yes, you just need to go to full screen.
Great, all right. Thank you, everyone. I apologize, my postdoc could not be here. He did most of the work. We asked a simple question, which was that could innate immune cells, like type 2 innate lymphoid cells, generate memory since they don't express antigen receptor like T cells or B cells? So we designed a protocol where we gave mice, RAG1 knockout mice, alternary allergens, 5 microgram per dose, 3 days a week for 3 weeks. And then we rested the mice for about 3 weeks, and then come back with a recall dose of allergen, which is 1/4 the sensitizing dose, which was 5 microgram, and the recall dose was 1.2 microgram. So it's a very low dose. And then we give 3 doses and look at the outcome 2 days later. So this is the ROA hyperreactivity measurement. You notice that the mice that were first went through the memory induction through alternaria and then received the recall challenge, developed ROA hyperreactivity. All the control groups are negative. So we clearly saw an asthma or ROA hyperreactivity response in this RAG knockout mice with the memory induction protocol. And this was associated with the eosinophils. Then we're interested in ILCs. So we look at the ICOS positive, ST2 positive ILCs, and they're significantly elevated compared to other groups. And this was associated with IL-5 and IL-13 protein in the bowel fluid. So just one flow cytogram to show the kind of visual impression of the ILC2s. This is your alternaria group, and these are all the controls. You can see significant increase in ICOS positive and ST2 positive ILCs in this model. I'm going to show you 2 additional slides. One is about single cell RNA-seq, and we did single cell RNA-seq in RAG knockout mice, looked at the hematopoietic cell, and these cluster of cells express all the genes that are expressed by ILC2s. So we analyzed the genes in this group, and we actually examine cells at 2 different time points: before the recall challenge, just after the memory induction, but before recall, and then also after the recall. So we'll have this 4 group, alternaria minus, meaning that memory induction without recall. This is the entire protocol and the control group. About 166 genes were upregulated in the memory model without recall, and 131 genes went down. Just briefly to give you an overview of what is the main story, we find 2 categories of genes involved in this memory formation. One category we saw increase in genes only after the entire protocol, shown here with the green bar. Another category of genes were increased after the memory induction, but they went down after the recall challenge. So there are a variety of genes in each category. I'm just showing 3 each. And then this is the attack seq of ILC2s. These are CD45 positive, CD25 positive, ICOS positive cells. This is perhaps the most important slide. So what you see here is just a memory without the recall, alternaria but no recall, and this is saline control. What is surprising and very intriguing is that gene access to genes were actually decreased after memory induction and across the entire genome. A group of genes showed only increased accessibility, but most genes were actually were less accessible compared to control. But then if you give these mice the recall channels, look at what happens on the right-hand side. They show overshoot, basically. They show a lot more gene accessibility after the recall challenge when a similar control group shown here did not have as many. So this is what we call the memory, the signature of memory. Initially during the memory induction, you will see repression of genes, and then, but they are prepared for a robust response, perhaps driven by this group of genes, and you see this robust response here. And some of the motifs that you observed that are driving this gene accessibility were primarily belonging to the AP1 complex, FOSL1, FOSL2, BAC2, and the preparedness type of molecules or motif were CREB-BF2, potentially BTF. And so this is the summary slide. We believe that repetitive exposure to a PAM-containing allergen, and we published in the JXMed why we think this is PAM and pattern recognition receptor-driven process. And we identified a new gene, memory-associated gene called FHL2 that drive ERK signaling, and it creates two genetic programs. One is the repression program, another one is a preparedness program. And we believe most of the repression is shown here by the ataxic and preparedness program, perhaps belonging to this group of genes. And then that's what creates the memory, which can be then elicited by the preparedness program and drives the phenotype of asthma.
Thank you very much, Dr. Alam. It's hard to get this significant of work into 3 minutes, so I appreciate your time.
Yeah.
We're going to move to the next speaker, and hopefully people can ask you questions in the chat, and we'll get to those in a second.
Our next speaker is Dr. Marijuan Hassani from UMC Utrecht, all the way from Netherlands. He will be speaking to us about the effect of mepolizumab on eosinophil and basophil kinetics in patients with eosinophilic asthma. Over to you, Dr. Hassani.
Thank you. To share my screen. There you go. Yeah, because of the limited time frame, unfortunately, I can only talk about part of the article that's already published in iScience. And today I'm only going to speak about eosinophil kinetics in blood. These are my disclosures, and the objective was to better understand eosinophil kinetics in humans after treatment with anti-IL-5. And to do so, we used the pulse chase labeling method, in which we labeled the DNA of eosinophil precursors with deuterated glucose. Deuterium is a stable isotope of hydrogen, and the patients, they had to take this label on day 0, they had to drink it. Patients had moderate or severe eosinophilic asthma, and 4 days after they took the label, they were treated either with mepolizumab or placebo. We enrolled 10 patients in each group, and we used FACS sorting to sort the cells of interest, or in this case, the eosinophils. We lysed the cells and we stored their DNA. And after we collected all samples, we used DNA hydrolysis to cut down the DNA into nucleotides. And we performed a chemical reaction to add the pentafluoride triacetate group to the nucleotides. And finally, we used GCMS to see which part of the DNA is actually labeled with deuterium. And then we can also see over time, because we collected blood samples on different days. So before I can show you the actual data, I have to explain it a bit. So what you will see in the graph Hereafter, on the x-axis the time, and on the y-axis the fraction of DNA, enriched DNA. And the first part of the graph to understand is the upslope part. This is the part where patients were drinking the deuterated glucose, and label is incorporated in the DNA of proliferating precursors, progenitors. So in this case, for eosinophils, the myeloblasts, the promyelocytes, and the myelocytes. But before the cells enter the blood, they need to mature or differentiate first. This is time that they do not proliferate, of course. We call it the post-mitotic transit time. And finally, there's a downslope part of the graph, which is actually the most difficult thing to understand, because it's not only loss of label due to cellular death or cell migration, but it could also be that there is entry of new label from the bone marrow, which is slower than the loss of label. So it can be the downslope determining factor. So now we can see the actual data, and first I can show you that if we look at the eosinophil count in the blood, after 2 days already, we see a very clear decline in the red group, which is the mepolizumab group, and this decline remains throughout the entire trial, while in the placebo group, the blue group, this is not seen. But if we zoom in to the kinetics data, you can appreciate that the first couple of days there's indeed no label in the blood measured. This is the post-mitotic pool transit time. It's around 4 days for eosinophils. This has been shown before. And then there's the up-labeling phase, which is similar in both groups, mepolizumab placebo group. This is not surprising because we gave mepolizumab after 4 days, so the actual label incorporation phase to start with was of course similar. But then the down labeling phase, that was different between the two groups. We saw that the downslope in mepolizumab was slower compared to placebo, and this was first surprising to us, but then we came up with multiple hypotheses that are not mutually exclusive. It could be that that the lack of IL-5 leads to accumulation of mature eosinophils in the bone marrow, so because they need IL-5 to be released from the bone marrow to the blood. So there's a gradual entry of label in the blood, which would then decline the downslope. It could also be that lack of IL-5 leads to a lower number of dividing progenitors. So we know that the proliferation rate is similar between two groups because we didn't see a difference in the upslope. But it could be that the amount of cells dividing at the same time could be lower in the mepolizumab group. Another hypothesis could be that the lack of IL-5 leads to accumulation of eosinophils in the blood because they don't have IL-5, they are not primed and they cannot enter the tissues. And finally, and I think that's the most interesting hypothesis that needs to still be tested, is that there might be multiple subsets of eosinophils, and then the IL-5 independent subset has different kinetics, slower kinetics compared to the IL-5 dependent inflammatory eosinophils. So to conclude, I think mepolizumab suppresses the number of proliferating precursors in the bone marrow, and it also suppresses mobilization of mature eosinophils to the blood and to the tissues, and possible that there's an IL-5 independent pool of eosinophils that has slower kinetics compared to the placebo group.
Thank you, Dr. Hassani. In the interest of time, I will have to stop you here. It was really an interesting talk, and I would ask Denise to go ahead and open the next talk, please. Thank you, Dr. Hassani.
Thank you. And for last but not least, we have Dr. Sina Karimi from ULB Brussels, going to be talking about mepolizumab efficacy and impact in patients with T-cell-driven hypereosinophilic syndrome, a biomarker sub-study in the recent placebo-controlled phase 3 clinical trial.
Hello, everyone. My name is Sina Karimi, and I'm going to be presenting a sub-study of the recent phase 3 trial of mepolizumab in HES, focusing on T-cell-driven disease. Quick background is that mepolizumab has been shown recently effective in uncontrolled HES. Next to that, LHES or the lymphocytic variant of Hypereosinophilic syndrome is a T-cell driven disease that comes with overproduction of IL-5 by subtypes of lymphocytes, specifically CD3 minus CD4 plus lymphocytes. And in the end, corticosteroid, there was a corticosteroid sparing trial before that showed a lower efficacy in LHES eventually, comparing to IHES for mepolizumab. Objects of this study was that to show if mepolizumab is effective in LHES, and also to investigate mepolizumab's effect on 3-4 plus T-cell subsets. What we did was to look at CBCs, do flow cytometry, do a couple of ELISA kits, and also look at T-cell clonality with samples that we received in the context of the phase 3 trial of mepolizumab in HES. The conditions of the study were the same as the phase 3 trial, and what we did was first to identify the patients with a T-cell disorder, then to analyze the study-defined outcomes for these patients, and then look at the influence of treatment arm on different biomarkers. First of all, we had 87 patients who consented to the study that were that their different characteristics were rather comparable in both treatment arms. As mentioned, first we looked at the different abnormalities of T cells, and afterwards, in the first part of the results, what we defined as a perfect outcome was absence of flares and an eosinophil count under 500 throughout the study. As you can see, no abnormality was significant and related to a lower chance of a perfect response. Although, as you can see in some of these groups, there were actually a small number of patients who did receive the active treatment. So this might actually explain the non-statistically significant character of these trends. Afterwards, looking at the placebo arm, we saw that patients with a CD3-4 plus phenotype and also patients with a clonal TCR do have a higher chance of having a flare if they do not receive any treatment comparing to other patients under placebo. Afterwards, we looked at the biomarkers, as I mentioned, for LHES patients, so 3-4 plus patients. The only difference was that patients under mepolizumab had an increase in their IL-5 levels comparing to placebo. And in patients without 3-4 plus phenotype, we did have a decrease in soluble CD25, so IL-2 receptor, in the active treatment arm, and also an increase in IL-5 levels among these patients. As I mentioned, IL-5 did increase in active treatment arms regardless of the patient group. And when we actually compared this increase, we did see that the increase was higher in patients with a 3-4 plus subsets. So they did increase their detectable IL-5 levels more with mepolizumab. Quick take-home messages. First of all, as I mentioned, for the moment, we can say mepolizumab is effective in LHES. Also, we saw a higher rate of flares in patients with a 3-4 plus subset. So they do have a severe disease if left untreated. And also, this study was not in part to assess the impact of treatment on 3-4 plus patients. So the open extension study would be an interesting moment to investigate that a bit more. In the end, IL-5 levels did increase as reported before, which is usually due to a complex of IL-5 and mepolizumab that is still detectable by ELISA kits. So our study suggests eventually these patients who increase their IL-5 more, they might have a higher production of IL-5. And in the end, the soluble CD25 levels decreased in active treatment, but only in LHES patients. In keeping with our previous in vitro observation that eosinophils contribute to activation of normal CD4 while inhibiting activation and CD25 expression by this specific subsets. Thank you very much.
Thank you very much, Dr. Karimi. I'm going to hand it over to Dr. Florence Rufos. We're going to skip our 3-minute question and answer and take those in the breakout rooms.
Hello, everybody. So the following 3 slides are going to show the titles of the talks that will be presented in our 3 breakout rooms. One of the breakout rooms are gastrointestinal disorders, as I said previously, and you have here the different presentations that will be presented as pre-recorded 1-minute, very, very brief presentations, and there will be ample time for discussion. The second breakout room is dedicated to, I don't know which of the 2 is going to come, but eosinophil biology. You can see here that there is a series of presentations that will also be pre-recorded 1-minute presentations, and once again, ample time for discussion. I'm just reminding you that the discussion here can be oral, and you can light up your videos as well as your microphones to directly interact with the presenters. The next slide shows the 3rd and final breakout room, which is Hypersinophilic Syndrome and other eosinophilic diseases. Once again, you have the 1-minute pre-recorded presentations, but in this room, you will also have other presentations. Oh, I'm sorry, it doesn't appear here, but the 3 last presentations that were not discussed will be also discussed in the relevant breakout rooms. Some of them in this one, but I realized that we have not updated this slide, if I'm not mistaken. So I'm sorry, you may have to try to find the speakers for those last presentations. So now it's time to break out.
That is still joining the rooms. I thought we would start at, well, 7:55 on my computer, 5 minutes to whichever hour time zone you're on. And Denise from IES is kindly going to load up our slides, and she'll run the videos, and then we should have lots of time for group discussions, at which point you can raise your hand, turn your camera on, ask the speakers their questions. In terms of, if you raise your hand, then I'll know to call on you, but if I don't see you, put it in the chat, or somebody feel free to tell me that I'm missing somebody's raised hand in case I can't see everybody on the screen. And also feel free to put your questions into the chat box to everyone, and then I can read those off for you. So I guess it is 5 minutes before the hour. We could go ahead and get started. So we think, Denise, whenever you have a chance to post the slides up, but I'll just go ahead and start introducing our speakers so we can get moving. So we've got 7 really nice talks coming up today, and this is the GI breakout room. I'm Sina Aceves. I am a pediatric allergist at University of California, San Diego and Rady Children's Hospital, and I work on EOE and mechanisms of remodeling. And it's really nice to see everybody this morning. Our first presenter is going to be, Denise, I don't know if there's a second slide that lists that. Thank you so much. So our first presenter is going to be Shmuel Avlas, and I'm going to apologize ahead of time if I mispronounce your name. Please forgive me for that. Who will be talking to us about the role of IL-13 alpha receptor as being critical in EOE pathogenesis. Our second presenter will be Nikhil Chauhan, and he is from the University of Illinois in Chicago, and he'll be speaking to us about a peptide antagonist that will block eosinophil recruitment to the esophagus. Our third presenter will be Nicola Dinny, and she is from the Francis Crick Institute, and she'll be talking about the aryl hydrocarbon receptor and how that promotes tissue adaptation of intestinal EOs using a mouse model. Kara Clearer will be our next speaker, and she'll talk about serum IgG4 in EOE, looking at 3 different studies. Our fifth presenter will be Sophie Albinson from University of Gothenburg, and she'll be speaking to us about T cells and eosinophils and how they accumulate together in T cell rich areas. And our next speaker will be Deborah Asano, and she will talk about the use of specific immunotherapy for allergic rhinitis in children who have EOE. And lastly, Alex Sugarman will talk to us about food impaction management in the emergency department. So with that, I think we can go ahead and get started. I don't know if it's just me, Denise. I can't hear any sound coming with the video.
Yeah, no sound.
No sound. Let me see if. Denise, we still can't hear it. I don't know if you can hear it on your computer. We can't hear it on our end. I wonder if it's because she's muted. Yes. Denise, I think if you unmute yourself and turn on your audio, we'll be able to hear it as well. Thank you. Always something new to learn with Zoom, even after a couple years. Thank you, Denise.
Still no sound.
I'm afraid we still can't hear it. Let's see if I could share my screen and see if we could get some sound up here. Denise, your screen shows that you're muted. So can you unmute yourself? I can't seem to force you to unmute. And then if you turn your output up to high, maybe that will help.
Our study on the role of IL-13 and the IL-13 receptor alpha-1 in our murine EOE model. In this work, we developed an experimental EOE model that results in increased tissue eosinophils, fibrosis, basal cell proliferation, and tissue thickening, as well as increased expression of hallmark type 2 associated genes and a significant overlap with the human EOE transcriptome. Using IL-13 deficient mice, we show that IL-13 receptor alpha-1 almost completely regulates experimental EOE. In addition, by bioinformatics analysis, we show the pathways that are being regulated by IL-13 receptor alpha-1. Next, we found that IL-4 does not play a significant role in the disease progression. However, inhibition of IL-13 protected the mice from experimental EOE. Therefore, we conclude that EOE is critically mediated by IL-13 signaling via IL-13 receptor alpha-1. Thank you very much.
Hello everyone, I'm pleased to share our research with you at the IES Virtual Forum. We identified a peptide from CCR3TM2 domain and modified to form nanoparticles. This inhibited chemotaxis of a CCR3 expressing cell line and mouse blood eosinophils, acting as a biased antagonist in beta-arrestin recruitment assays. Peptide also reduced eosinophils in the lungs, airways, and blood in a mouse asthma model. We hypothesized this peptide could also be a potential therapeutic in eosinophilic esophagitis. Day 0, mice were skin sensitized with oxalone. Days 5, 8, 10, and 12, mice were injected IV with peptides at 12 mg per kg and orally challenged with oxa. Day 13, we sacrificed the mice, collected the esophagus for ISC to determine the number of eosinophils. Results showed significantly eosinophil infiltration into the distal, middle, and proximal esophagus. Importantly, distal epithelium and lamina propria showed significantly reduced number of eosinophils and reduction in abscesses and surface layering in the CCR3 peptide treated group. Our preclinical EOE model suggests the CCR3 peptide antagonist as a potential therapeutic to investigate in further studies. Thank you.
The aryl hydrocarbon receptor is a ligand-activated transcription factor with important roles in gut physiology. We identified HR as a critical factor in mediating tissue adaptation of eosinophils in the intestine. Comparing intestinal to bone marrow eosinophils revealed large transcriptional changes, including pathways involved in cell-cell interaction, extracellular matrix remodeling, and immune response genes. The HR pathway was highly upregulated in intestinal eosinophils. We confirmed high HR expression transcriptionally and in reporter mice, and showed that eosinophils upregulate HR after migration into the intestine. HR-deficient eosinophils showed altered granularity and survival, and RNA sequencing revealed that HR controls many of the pathways normally upregulated in intestinal eosinophils. Thus, the HR controls part of the tissue adaptation program of intestinal eosinophils in mice.
Here we show results from 3 studies of serious chronic diseases such as CKD.
That's true. If there are any other questions on the CKD study, feel free to put them in the chat. In the meantime, there was a question for the COVID HES study. This is for David Espinoza. Does the assessment of COVID infection rates in HES suggest that eosinophils are not playing an essential role in respiratory viral infections?
That's a good question. I don't think it says that they're not playing an essential role, but there's a lot of other confounding factors that we have to take into account. So I don't think we can say for certain that they're not playing any role in respiratory viral infections.
Right, and I think, at least for the HES patients that are included in that cohort, they have a variety of manifestations, not just, respiratory. So I think all it really tells us is that there's not, a big role for either reduction of the eosinophils or the disease itself in making more, COVID severity in these patients. So Dr. Rufus has one more question.
Yes, hello. This is a question for the eosinophilic disease overlap abstract. The slides went very rapidly. 1 minute is really hard just to capture all the information that's out there. So I was wondering perhaps if you could explain more how you looked at this, what exactly did you enter in terms of search terms, what were the actual overlaps that you were interested in looking at?
25%. We saw 25% overlap between human disease and mouse disease. I hope this answers your question.
Yes, it does. Thank you. It was a very nice presentation, by the way.
Thank you.
I think Denise, you might be back on. Maybe we lost you for a little bit. We lost the presentations after the aryl hydrocarbon receptor talk. So maybe if you give me a heads up before you want to start. Are there other questions people want to ask right now? I have a question about your IL-13 mouse as well. I was wondering, you see a lot of lamina propria fibrosis and remodeling in your model, and it's really lovely because there's not that many models out there that are super robust. And I was wondering, with your IL-13 blockade and with your IL-13 knockout mouse, if you think that the effect on fibrosis is directly through IL-13 or via the immune cells based on the gene enhancement signals that you see?
Well, I really need to study it more, but I think it's more related to the IL-13 receptor alpha-1. This is where we saw the most robust response. And as much as I know, IL-13 receptor alpha-1 expressed more on epithelial cells. So I think it's the receptor that played a major role rather than the immune cells.
I also had a quick question. I mean, in the model, again, I'm not too familiar with the model, but
just wanted to know whether there is any features which is characteristic of the human EOE. So, for example, whether there's any strictures and whether there's some food impaction, etc., which is mimicked by this model.
Well, in this model, I did not see significantly stricture and food impaction. Of course, I saw in some mice esophagus food stricture and food impaction, but it's not significant. So in this case, the answer will be no.
And just a follow-up question. You mentioned obviously there's some correlation in terms of the genes between human and obviously the genes upregulated or modulated in this model. Do they link to specific pathways which are very similar, or have you looked at some kind of pathway analysis, associated with those genes?
So what we saw that is overlap, of course, we have the list of genes, but we saw clusters. And the clusters that are overlap are the immune response and proliferation.
Thank you.
This is Stephen Ackerman again. Did the model show abscess formation or eosinophil surface layering? And if so, did those things change differentially with blocking IL-13 receptor? Were there any effects that were kind of different, that might have been different? So I guess I'm trying to get at whether or not, if IL-13 is a major mediator in terms of the transcriptome and what the tissue looks like, are there any differential effects on some of those kinds of responses? Kind of a follow-up to Sima Sevi's question.
So if I get it right, to answer your question, we did see some eosinophil layering, and in some mice we also see abscess. When we use the knockout mice, the mice were completely not sick at all. They looked like the control group. And when we block the IL-13 cytokine, of course, we saw effect, but it's not like the blockade of the receptor itself.
Thank you very much. Denise is back on, so I'm gonna. It was a lovely model, Shmuelk. Denise, I'm gonna go ahead and let you restart so that we can see the rest of the talks. Whenever you're ready, go ahead. Sorry, Denise, we can't hear.
Higher in milk elimination diet responders compared to non-responders. BOS-D8 showed this trend as well. Levels of BOS-D4 decreased after milk elimination. Levels of BOS-4 and after elimination. Aim was to investigate serum IgG. We show results from 3 studies of serum IgG4 titers in eosinophilic esophagitis. Our aim was to investigate serum IgG4 antibodies to cow's milk proteins in EOE. In adults and children, we measured serum milk protein IgG4 titers before and after eliminating milk from the diet. Histologic response to milk elimination was also assessed. We found that prior to milk elimination, levels of BOS-D4 and BOS-D5 were higher in milk elimination diet responders compared to non-responders. BOS-D8 showed this trend as well. Levels of BOS-D4, D5, and D8 decreased after milk elimination. The reduction was 15 to 20% higher in diet responders compared to non-responders, although not statistically significant. We conclude that elevated serum IgG4 levels to major milk proteins are associated with response to milk elimination in EOE.
My name is Sophie, and I will present to you my recent work on eosinophils in the esophageal mucosa of patients with eosinophilic esophagitis. We have investigated esophageal biopsies from 20 patients using immunohistochemistry. Our main finding was that the eosinophils were accumulated in T-cell-rich areas of the esophagus, and that the eosinophil numbers in the esophagus correlated with both CD4 and CD8 T-cell numbers. We also investigated CD16 positive eosinophils, which are a subgroup of eosinophils with increased ability for T-cell suppression, and saw that the percentage of these CD16 positive eosinophils decrease after successful treatment. So what we see is that there is a relationship between the eosinophils and T cells, and we hypothesize that the eosinophils in the esophagus of eosinophilic esophagitis patients suppress T cells. Thank you.
Good morning. The title of my project is as follows. Our two aims were to determine the rate of allergy shot therapy in EOE new diagnoses and to determine the association between PPI use and said therapy. We hypothesized that EOE patients receiving allergy shots suggestive of severe AR would have higher rates of PPI use. We performed a retrospective chart review of surveys completed in the Egypt clinic to perform the study. For our results, only 5.7% of patients studied received allergy shots at least 3 months prior to EOE diagnosis. Overall, 62.8% of patients used PPI. Patients who had allergy shots were 3.6 times more likely to have had PPI prior to EOE diagnosis than those who did not have allergy shots. Below shows adjusted odds ratios for race, ethnicity, and sex. In conclusion, a small minority of EOE patients used allergy shots and more were on PPIs. Additionally, uncontrolled AR and allergy shots were associated with PPI therapy. Further studies are needed to determine whether these associations are due to esophageal dysfunction or GERD.
Hi everyone. Data shows that nearly 1/3 of patients with eosinophilic esophagitis, or EOE, experience one or more food impaction. Yet no standard protocol exists for managing food impactions in this patient population. So we created the EOE Food Impaction Emergency Care Plan via PubMed review of best practices. We surveyed ED physicians and patients with EOE to assess perceived value and comprehensiveness of the proposed emergency care plan. Notable findings include, with this care plan, 87% of patients would feel somewhat or significantly more comfortable seeking care during an acute event. 75% of physicians responded no or not sure regarding awareness of an existing standard treatment protocol, and 60% of physicians and 87% of patients somewhat or strongly agreed that the proposed care plan is comprehensive in its recommendations. The survey was also useful in determining areas for improvement on the content and organization of the proposed care plan. Scan the before and after QR codes to view the original version and the updated version incorporating participant feedback. We also recognize the limitations of this study's scope. Further studies should include a large sample of ED physicians and patients, as well as input from other stakeholders such as gastroenterologists, endoscopists, and nurses.
Okay, very good. Thank you so much. We probably have time, hopefully, for a couple of questions. There's one in the chat from Ava. Question for Sophie. Do you notice CD16 positive EOs suppress all T cells equally, or do they suppress any subset, such as CD4, 8, or Tregs more than others?
Yeah, that is actually a really interesting question. Sadly, we have only looked at CD3 positive T cells so far, but I will definitely take this as a new thing to look at.
Perfect. I think we have about a minute left. If anybody has a quick question, please speak up. And if not, maybe Nikhil, I'll just ask you a quick question. Did you see any change in the activation state of the eosinophils? I know the numbers were reduced. Was the activation state changed as well?
We haven't looked really for the activation. Yeah, but we were just only for the focus only the number of eosinophils infiltrated to the esophagus for now. Yeah, but we can look into that in future.
Very good.
Yeah, we plan that for the future, Seema.
Beautiful. Really nice. Anybody else have anything quick they want to jump in with? Please do. No?
Hello, everybody. We're in the process of having everybody join the breakout rooms. There were a tiny technical glitch of people trying to join. So if you can give us a second, and then once we get our moderators in the room, we can start the videos. Looks like more people are joining. So I think we're ready. We have a critical mass. If we can go ahead and roll the slides. And so just to mention, these were the speakers that are going to be presenting today. These are all pre-recorded talks. We can go ahead and start playing.
Hello, I'd like to present this abstract on behalf of the co-authors. The abstract is entitled Lirentelimab, a monoclonal antibody against cyclic 8 depletes blood and tissue eosinophils in patients with allergic or inflammatory diseases. My disclosures are indicated on this slide. Lirentelimab targets cyclic 8 on eosinophils and mast cells. The drug has now been studied in 4 clinical trials, including over 150 patients. These studies include the recent ENIGMA study, which was focused on eosinophil gastritis and eosinophil duodenitis with 58 patients. Collectively, the data about the effect of this drug on blood eosinophils is shown in this slide. And one can see very dramatic decreases in blood eosinophilia that was sustained during the drug treatment and after the drug was discontinued. Focusing on the data from the ENIGMA trial, one can see the effects on the histological remission. This is defined by the levels of eosinophils being less than or equal to 4 in the stomach and less than or equal to 15 in duodenum. And one can see that over 94% of the patients achieved this strong remission at the various time points studied, going out to date 659. So in summary, lirentelimab depleted blood eosinophils in 4 separate studies of patients with allergic or inflammatory diseases through 24 weeks. In patients with EG and ED, monthly dosing of lirentelimab maintained complete or almost complete depletion of blood and tissue eosinophils through 94 weeks. Ongoing trials with lirentelimab are focused on phase 3 and phase 2 studies related to EOE, EG, and EOD. Thank you for your attention.
Hypereosinophilic syndrome is a rare heterogeneous disorder characterized by an absolute eosinophilic count of greater than 1,500 cells per microliter with either tissue involvement or organ dysfunction. Eosinophilic hepatitis is a rare manifestation of hypereosinophilic syndrome that has not been well described. The clinical, radiographic, and histologic findings of 12 patients with hypereosinophilic syndrome with hepatic involvement were reviewed. 3 patterns of injury emerged. Inflammatory, evidenced by inflammatory cell infiltration, primarily eosinophils, with various forms of hepatocellular degeneration or necrosis, vasculopathic, in which the vasculature was primarily affected, and a mixed pattern in which there was inflammation of the liver parenchyma and bile ducts. This figure depicts the clinical course of one patient with an inflammatory presentation. As you can see, with the initiation of glucocorticoid therapy during times of flares, there was significant improvement in the clinical and biochemical parameters. Thank you.
Hi, I'm Dr. Mark Rothenberg, and I'm one of the chief lead authors on this study entitled Impact of Baseline Blood Eosinophil Count on Flare Reduction in Mepolizumab-Treated Patients with Hypereosinophilic Syndrome. Disclosures are indicated. This study was a subgroup analysis in previously reported data, which showed that mepolizumab compared to placebo was effective in a phase 3 trial of 12 and over patients with hypereosinophilic syndrome. In this analysis, we are examining the impact of the baseline blood eosinophil count on the outcome of the study, which is flare reduction. The data is primarily shown in this slide and basically shows that the drug versus the placebo has a dramatic effect irregardless of the blood eosinophil count. The data is shown as a bar graph on the left, and as a rate ratio of the baseline blood eosinophil count as a function of the different response rates for the drug versus the placebo on the right. So in summary, these results indicate that patients with HES that are not FIP1-PDGR alpha positive are likely to benefit from mepolizumab treatment, irrespective of the baseline blood eosinophil count. Thank you.
Hello, my name is David Espinoza, and I'm a postdoc at the NIH in the Human Eosinophil section. I'll be presenting my project, COVID-19 Infection and Hypereosinophilic Syndromes, a Survey-Based Analysis. Hypereosinophilic syndromes, or HES, are a group of rare disorders defined by elevated peripheral blood eosinophils and eosinophil-related disease manifestations. Our project aimed to better understand infections and potential complications of COVID-19 in patients with HES. Participants were enrolled in 2 rounds of REDCap surveys with collection of demographics, HES disease status, medications, and COVID-19 exposure. We identified that 17 of 158 unique HES patients tested positive for SARS-CoV-2. This cohort of COVID-positive patients were comparable in demographics and HES status to the overall HES cohort. The hospitalization rate was similar to the 21% rate reported by the CDC on March 2020 for individuals who tested positive for SARS-CoV-2, but still higher than the 9% hospitalization rate reported for individuals with no underlying health condition. In summary, our data shows that there is no enhanced susceptibility or severity of COVID-19 to date in this cohort of patients with HES, and we are still looking into vaccine information.
This is a large real-world retrospective study evaluating the association between blood eosinophil levels and adverse outcomes in patients with chronic kidney disease in stage 3 or 4. Optum US electronic health records data were analyzed after adjusting for clinical and demographic characteristics, as well as other known risk factors for CKD in different disease stages stratified by different blood eosinophil ranges. Here are the results of the study. Hazard ratio in Cox regression analysis indicate that the patient with CKD in stage 3 or 4 having blood eosinophils above 300 are more likely to progress compared to those with normal eosinophil levels. Also, Kaplan-Meier curves show that patients with increasing levels of eosinophil progress faster. In conclusion, this study indicates that the risk of CKD progression and all-cause mortality was higher in patients with elevated blood eosinophil levels independent of known risk factor, suggesting that blood eosinophils may be used as biomarker of CKD progression.
This is a study on the real-world investigation of eosinophilic-associated disease overlap REVEAL. The REVEAL study aimed to describe and characterize disease overlap in terms of patient demographics, disease severity, eosinophil levels, healthcare resource utilization, and costs. We used data from Optum claims between 2014 and 2019. Eligible patients had at least 2 years of continuous health plan enrollment and at least 1 out of 11 eosinophilic disease diagnoses of interest. Higher eosinophilic disease overlap rates were observed in patients with severe disease compared to those with mild to moderate manifestations of the disease. Figure 1 shows an example on eosinophilic esophagitis and eosinophilic granulomatosis with polyangiitis. We also found that patients with overlapping conditions had increased healthcare resource utilization compared with patients without overlapping diseases, as shown in Figure 2. Higher rates of eosinophilic disease overlap were associated with older age, higher eosinophil levels, and higher costs, results not shown. In conclusion, this study informs on the need for healthcare providers to query for overlapping eosinophilic diseases when assessing patients.
The majority of patients with lymphocytic variant Hypersinophilic syndrome present with continuous and soft tissue manifestation, while cardiac complications are extremely uncommon. We recently observed severe vascular complications in 2 patients that we have been following for more than 10 years and whose clinical presentation were very benign until then. Patient 1 was a 52-year-old female who experienced a stroke with right facio-brachial paresis and non-fluent aphasia. This was due to spontaneous dissection of the left middle cerebral artery. Patient 2 was a 62-year-old female who developed an acute myocardial infarction due to dissection of the proximal ascending aorta and development of large pseudoaneurysm that contained neurothrombin and compressed the left coronary artery. Neither patient had cardiovascular risk factors, and both patients had persistent hypereosinophilia over the years preceding the acute event. Development of vascular aneurysms in the setting of otherwise relatively stable and benign LHES has been reported in 3 other patients. Although this is a very uncommon disease, complications and its consequences are very serious.
Hello.
And I believe that is all. So at this point, just in the interest of time, I don't see a lot of questions posted in the chat, but feel free to come off mute and ask your questions directly. There's a lot of interesting talk, and you can also raise your hand as Bhavini has done. So I will go ahead and let you unmute yourself and share your video if you'd like and ask a question. Unless that was a mistake. Okay, Dr. Rufos, go ahead.
Thank you. I was just wondering why, what's the reason that lerontinemab is mostly being assessed in gastrointestinal disease? Is there some kind of rationale behind that approach, or what are the plans in terms of development?
So I believe this question is for Dr. Rothenberg, or
yeah, this is a good question, and it's more of a business decision by the company. So I'll let Dr. Singh respond.
So Dr. Bhupinder Singh appears to be on the line too. I don't know if you'd like to take that question, or we can move on. Okay, I believe we don't have an answer for that question, so I'm going to move on to Dr. Amy Cleon. If you'd like to unmute yourself, share your video if you'd like, and ask a question.
I'm unmuted, but I'm not sharing my video because I'm driving. But I have a question for the person who spoke about the renal disease. Do you know what impact treatment has on that? So if you treat the eosinophilia and you lower it, does that improve the prognosis in chronic kidney disease?
Hi, Ami. We didn't look at the impact of the treatment on any of the CKD stages, because this was based on the data claims analysis. So this was not really possible to perform this type of analysis.
Thank you very much. Are you aware of anyone actually looking at that moving forward?
Not as far as I know. I think this is a good idea for any more controlled trials, prospective study to look if there's any impact on treatment, on the risk of progression, and in relation to eosinophils levels in those patients.
Yeah, I mean, it's a problem with the clinical trials because many of them exclude people that have creatinine clearance below a certain amount. So obviously it would be interesting to look perhaps in a cohort of patients that are not being treated in a clinical trial, or that's being treated specifically for this reason.
Yes, we were thinking about this as well, but it's it's difficult to find a database, and the patient who were on biologic or treated with steroids, for instance, if there's any impact. Yeah, it's a good idea to look at.
Yeah, I think it's interesting. The counts themselves are not terribly high. They're just, different in terms of the baseline counts.
But look, we look at the blood eosinophils level. So as I see the other people presenting here, like TBC, and I think there is the eosinophilic driven inflammation in this in the many chronic diseases such as CKD.
That's true. So, if there are any other questions on the CKD study, feel free to put them in the chat. In the meantime, there was a question for the COVID HES study. This is for David Espinoza. Does the assessment of COVID infection rates in HES suggest that eosinophils are not playing an essential role in respiratory viral infections?
That's a good question. I don't think it says that they're not playing an essential role, but there's a lot of other confounding factors that we have to take into account. So I don't think we can say for certain that they're not playing any role in respiratory viral infections.
Right, and I think, at least for the HES patients that are included in that cohort, they have a variety of manifestations, not just, respiratory. So I think all it really tells us is that there's not, a big role for either reduction of the eosinophils or the disease itself in making more, COVID severity in these patients. So Dr. Rufus has one more question.
Yes, hello. This is a question for the eosinophilic disease overlap abstract. The slides went very rapidly. 1 minute is really hard just to capture all the information that's out there. So I was wondering perhaps if you could explain more how you looked at this. What exactly did you enter in terms of search terms? What were these actual overlaps that you were interested in looking at?
Yes, can you hear me?
Yep.
So we looked at the overlap of 11 eosinophilic diseases: COPD, asthma, atopic dermatitis, bullous pemphigoid, non-cystic fibrosis, bronchiectasis, eosinophilic gastritis, gastroenteritis, EGPA, eosinophilic granulomatosis with polyangiitis, eosinophilic esophagitis, COPD, and chronic spontaneous urticaria. So we used the Optum claims to look at this, and we aimed to characterize disease overlap, so to see which diseases co-occur in which kind of clusters, in which kind of sequences, whether certain clusters are associated with higher disease severity, if patients with several comorbid diseases have higher eosinophil levels, for instance, and then also characterizing those disease clusters in terms of healthcare resource utilization and costs.
Mm-hmm.
So I think the implications are to encourage healthcare practitioners perhaps to look at those disorders not as standalone conditions, but as disease clusters, and to understand also the role of eosinophilic immune dysfunction in driving the development of several related conditions, and to try to...
And are there any perspectives in terms of looking at genomics and seeing if patients who have multiple eosinophilic conditions actually somehow have a predisposition to develop type 2 or eosinophilic diseases? Are there
That's
any?
an interesting question. We haven't considered that. I think it's a little bit too fundamental research question for me. I would have to check with my team. I'm an epidemiologist, but yeah, it would be interesting maybe to take it forward.
Florence, this is Danuta. I'm also involved in this study. I think this would be, it's very interesting, a really research topic, but this would require the blood for those patients who have with multiple conditions. So then we can really see, we can have some, collaboration on this. But I think it's very, right now with this database, I don't think it will be possible.
Okay, thank you.
I think we have another question from Dr. Amy Cleon.
Yeah, so one of the 3-minute talks was on the FIP1L1 PDGFRA and watershed infarct. Is that in this session?
It should be. It should be.
Should be. We hope that they join.
All right. Well,
yeah,
cool.
yes, I am here.
Thank you.
Okay.
Yes.
Good. So I had a question. So do you know, oftentimes what happens is a patient has FIP1L1 PDGFRA, and because it's treatable with imatinib, the evaluation stops there. But do you know if NGS or whole genome or something was done on those patients, and whether there are other genetic factors that might predispose to watershed infarct in those patients?
Thank you for the question. So these NGS explorations are not performed routinely. So it wasn't done for these patients. But what we can say is that these watershed infarcts are shared with many eosinophilic syndromes that are clonal, but also secondary. So we're not sure. We might find something, but I can't answer because we haven't performed it.
Thanks. I mean, the reason I ask is so when you do, we've found that some of the patients, not all of them, when there's been a very extensive coagulation workup done in our patients who have these kinds of strokes with normal cardiac, normal cardiac studies, that it seems that I can't tell you that it's statistically anything. It seems that abnormalities in the coagulation pathway may be more prevalent. And so it may be that the combination of very high levels of activated eosinophils plus something else leads to infarct. And so I think it's an interesting, you have a very interesting group. There probably is DNA and RNA stored on these patients because the diagnosis of 1.0 and PDGFRA was made. So it might be a very similar otherwise cohort to look at to try to see if this can be dissected out.
Yes, indeed. I will report to my to my team to see if we could go further into details on the DNA samples. Thank you for your suggestion.
Thank you. And before we go to your question, Dr. Rufus, I'm going to ask a question of you and your group. Can you maybe think about or explain perhaps your thoughts on the cause for vascular dissection in the patients with LHES?
So to be perfectly honest, we're looking into it and we don't know. Obviously, the first thing that we wanted to look into was to see if there was a presence of eosinophils, as has been described in one of the 3 cases that have been published so far, around or in the vascular wall. And we actually sent some material because the second patient had a major cardiac surgery with valve replacement of the proximal aorta, coronary implantation. So all that tissue was available, and we sent it to Chris Leiferman in the States to look for granule proteins. And the problem is they had no control tissue for that. And so although they did seem to see some staining, they're not ready to really formally say that this was perhaps due to the presence of eosinophils. What was surprising in both cases is these were patients who had very, very, very mild disease manifestations. One was not even treated because she actually had no disease manifestations for the previous 10 years. And so we can only hypothesize that this has to do with eosinophils, and it has been described rarely in exactly the same setting.
Thank you. And you can go ahead and ask your question now.
Thank you. Well, it was also a question for Juliette Tenenbaum on their cohort of patients. I was wondering so these were clearly patients, the majority of which the stroke was the actual inaugural symptom of their disease, if I understand. And I was just wondering if there were any differences in eosinophil counts at diagnosis in this subset compared to the subset of patients who did not have stroke.
Thank you for the question. So there was no significant difference between eosinophil blood count at diagnosis, but we have a tendency. Patients with stroke had a 12.8 giga per liter of eosinophils, whereas patients without stroke had 8 giga per liter. So it was not significant, but maybe there's a tendency for higher eosinophil blood count. However, we couldn't conclude that. And regarding other blood laboratory findings, there was no difference between the 2 groups.
Thank you. I was asking that question because it's kind of related to our short abstract. Yes. We saw a very, very sudden and marked increase in eosinophil counts at the time our patients developed these vascular complications. And it intrigued, it's only 2 cases, but it was intriguing to us.
Yes, indeed. Thank you for your question.
Okay, I think we have just 1 more minute for any last burning questions. Otherwise, I think we may just, have a little chat. I think one of the things that we miss about the International Eosinophil Society in person is that we actually meet all of our collaborators and make lots of contacts. So if anybody would like to just say hello and show their video, that would be great. If not, I'll just stay here and you can just look at me. I think,
which is nice.
Thanks, Florence. Yeah. Hi, Mark. How are you? It's nice to see you. These were all really nice talks, and hopefully we'll all get to reconvene back in person fairly soon, hopefully.
So, Denise, do you know if the others, so some of us feel torn between the basic science and eosinophil biology and clinical work. Are these sessions recorded, and are we going to be able to watch them at a later date?
Well, I
They
think
are
so.
recorded.
It does recording. Yeah,
Yeah, they're
so you might
recorded.
be able to see the other sessions too.
Yeah. But as I'm going to announce just afterwards, we're really going to be asking for feedback from the attendees on improvements that should be made based on frustrations like the one that you just voiced.
Florence, my frustration was 1 minute
Yeah,
presentation.
like everyone. So
but
maybe
it was excellent. Thank you very much.
Denise, if we just leave the room, will we all go back to the main place, do you think? Do we dare to push on that little button?
I think you can choose breakout room and go somewhere else actually. If you hit the breakout room button, you can actually go somewhere else. So
the breakout room button.
I think at some point somebody will close all the rooms and we'll all end up back in the main room. So, so thank you very much, everybody. This was really fun. And thanks for answering all the questions to the presenters, and see you all back on the other side.
Thank you, Denise.
Thank you very much.
Also analyzed eosinophils under pathologic conditions. Knocking out ATG5 in the eosinophil lineage resulted in delayed and reduced eosinophil differentiation under both physiologic and pathologic conditions. As a result, we observed accumulation of immature eosinophils in the bone marrow and a significant drop of mature eosinophils in the circulation. Moreover, ATG5 knockout mouse eosinophils and ATG5 low-expressing human eosinophils exhibited enhanced degranulation abilities, suggesting that eosinophil effector functions are negatively influenced by ATG5-dependent autophagy. Taken together, we show that mouse and human eosinophil hematopoiesis and effector functions are regulated by ATG5, which controls the amplitude of overall eosinophil immune responses.
Hello everyone, my name is Avishai and I'm a PhD student from Professor Munitz's lab at Tel Aviv University. Today I'm going to present you my project about transcriptomal profiling of eosinophils polarization. First, as we all know, eosinophils are classically related to type 2 diseases such as parasitic infection and allergies. In recent years, it was found new roles for eosinophils, associated them with type 1 environments, such as combating microbes in colon infections. From other myeloid cells, we know that exposure to polarized environment leads to polarization with unique phenotype. So, we were interested to define this polarization state in eosinophils. Thus, we activated eosinophils with classical type 1 and type 2 stimuli. We found, as can be seen by PCA plot, that this stimulation resulted in significant transcriptomic changes in eosinophils with distinct phenotype. The greatest difference was observed between IL-4 stimulated eosinophils to eosinophils which were stimulated with interferon gamma plus E. coli. Finally, by bioinformatics analysis, we discovered that different stimuli induce distinct pathways. Thank you.
Hi, my name is Josh Mitchell, and I'm a graduate student in Dean Mosher's lab at the University of Wisconsin-Madison. We are interested in determining the differential cellular processes that commit eosinophils to terminal endpoints after activation with the cytokines IL-5 and IL-33. Laura Muehlbauer, a collaborator from Josh Kuhn's lab at UW-Madison, used multiplex tandem mass tagging to compare phosphocytes in human eosinophils treated for 10 minutes with IL-5 versus nothing, IL-33 versus nothing, or IL-5 versus IL-33. Each comparison was made on 5 pairs of samples, giving us great power to find differences. Roughly 36,000 sites were identified, with approximately 9,500 significantly changed sites. Upon cytokine activation, a set of common biological processes, cytoskeletal rearrangement, vesicular trafficking, ubiquitination, and splicing, were all upregulated. IL-5 activation resulted in upregulation of JAK-STAT signaling, and IL-33 activation led to upregulation of NF-kappa B signaling. These datasets give us a detailed glimpse into the biological pathways required for general activation of eosinophils, as well as the pathways that commit eosinophils to IL-5 and IL-33 activated states. Thank you.
Hi everyone, my name is Timothy Fetrelet and I'm a PhD student under the supervision of Professor Hans-Uwe Simon and Professor Shida Yosefi at the Institute of Pharmacology at the University of Bern in Switzerland. Today, I would like to share a recent report where we studied the link between eosinophil degranulation and mitochondrial DNA release within the process of eosinophil extracellular trap formation. We investigated the time-dependent release of the granule protein EPX and mtDNA following C5s stimulation of both human and mouse cytokine-primed eosinophils. We observed the maximal EPX release and upregulation of the surrogate marker C63 already after 1 minute of stimulation, while mtDNA release only reached a maximal level after 30 to 60 minutes. These findings suggest that the molecular pathways leading to degranulation and mtDNA release are at least partially different, and that the association between granule proteins and mtDNA scaffold is likely to occur in the outer space. Thank you for your attention, and I look forward to discussing with you in the breakout room.
Hello, my name is Yash Choksi, and the title of my poster is Eosinophils May Be Protective in the Development of Murine ESCC and Alter the Tumor Microenvironment. In this poster, we demonstrate that eosinophils are present within areas of human ESCC and in a significantly greater number in early stage versus late stage disease. We then show the same is true in a 4-nitroquinolone 1-oxide murine model of ESCC. Next, we determine that modifying eosinophil number, either with an anti-IL-5 monoclonal antibody or in an eotaxin 1 knockout mouse, resulted in significantly worse disease. Next, we showed that in mice treated with an anti-IL-5 monoclonal antibody, there was not only worse disease, but also a significantly greater number of CD4 positive T cells. Finally, we showed that in vitro co-culturing an eosinophil cell line, or EOL1 cells, with cancer cells resulted in a significantly greater apoptosis. I look forward to discussing future directions or any questions any of you might have about my work. Thank you so much.
Today, I'm reporting inflammatory and regulatory eosinophil gene expression in an in vitro model of eosinophilic esophagitis. Eosinophils have pleiotropic functions in tissue environments and may adapt regulatory or inflammatory phenotypes. Outstanding questions in this area include what roles different phenotypes play in inflammation, what factors promote differentiation, and what changes define that differentiation. We examined gene expression by circulating eosinophils and eosinophils co-cultured with esophageal epithelial cells with and without IL-13. Of the published inflammatory eosinophil genes, the majority were upregulated in co-culture and to an even larger extent with IL-13 supplementation. By contrast, regulatory eosinophil genes were by and large detected at the lowest level in IL-13 supplemented co-culture. Evidently, eosinophil gene expression is highly responsive to the local cytokine environment and may adapt an inflammatory phenotype in IL-13 supplemented co-cultures. Future studies will assess whether eosinophil functions such as cytokine production change in these conditions.
Conflicting data has emerged regarding a role for eosinophils in IgA production, with some reports that eosinophils support IgA levels during homeostasis. In my study, where wild-type and eosinophil-deficient double-gata mice were purchased from the same vendor and bred as littermate controls, I found that eosinophils are not required to maintain homeostatic levels of secretory IgA. When I went on to measure circulating antibody isotypes in these mice, an interesting phenomenon emerged. When these genotypes were born from separate dams and either co-housed as adults or co-reared their entire life in the same cage, I consistently found a significantly elevated level of circulating antibody isotypes in double-gata mice. But when I repeated these assays in littermate controls, the antibody levels were always the same. Together, this indicates that rearing conditions can impact steady-state antibody phenotypes and emphasizes the importance of optimally controlling rearing and housing conditions throughout life between mice with different genetic backgrounds.
I think we have completed our 7 abstracts for the breakout room. I thank Nina Jermick, Avishek Dolitsky, Josh Mitchell, Timothy Frettelet, Yash Choksi, Julia Dunn, and Rachel Fitzpatrick for their teasers, 1-minute teasers into their research. It was fascinating. Thank you so much. I'm going to open the floor for discussion. It can be in any phase or format. We have about 10 minutes for discussions. Thank you. Any burning questions? Anybody? Please feel free to unmute yourself and ask the question to the presenters themselves. Anybody? Okay, so I have a question for Julia Dunn. Do I have Julia Dunn on the, yes, I do see her. Julia, you presented, it was a very interesting one. You presented a gene expression profile, and you said there were regulatory genes for eosinophils that were suppressed on IL-13 co-culture. Which specific genes were you talking about? Sorry, it just went in a flash, that's why.
Yes, that is, so it was in the text of the abstract, and I had thought that, but let me pull that up. So yeah, not all of them were downregulated. It was actually really, it's interesting, kind of the mixed phenotype. But it was sort of the same for the inflammatory genes. A lot of them are really high in the co-culture with IL-13, but a lot of them were up at baseline. But in terms of the regulatory genes that were down with the co-culture plus IL-13, some of them were RUNX3, IL-17 receptor. SELL is a big one, and one that we've been looking at the protein level, because it's pretty straightforward to just, it's a surface marker so that we can measure it by flow, and we have been recapitulating that. EMR1, CCR3, or just a couple of the other genes that were that have also been sort of reported to be associated with the regulatory phenotype that we saw as being downregulated in our model. But again, all of that information about, what really defines a regulatory and an inflammatory eosinophil is, it's sparse. I think that these words have been used a lot, but in terms of, and also, kind of what are we looking at in a mouse versus a human eosinophil? So I think a lot of this is up for grabs. And if my real feeling is that there's more than two flavors, and we're really just kind of scratching the surface.
That might be the scenario. There's been a recent paper that just came out in JCI by Dr. Anuradhare and Dr. Sally Wenzel looking at IL-18 in severe asthma. Did IL-18 come up as one of the genes that you investigated that may be affected by IL-13 environment or the epithelium?
It's not in this particular kind of sub-study, no, but that is something that I had been looking at a lot. It came up as one of the cytokines, actually one of the only cytokines that was specifically upregulated in our co-cultures. I think was actually more likely to be produced by the epithelial cells, just based on the RNA-seq data. The eosinophils were differentially expressing receptor subunits. And so I do think that IL-18 signaling is involved, but I'm not sure about the eosinophils like producing IL-18.
And it was the co-culture with the epithelium, right?
Yes.
Right. Okay, I've hogged enough of the space. Anybody, please? Questions? Any question to Yash Choksi or Josh Mitchell? That was some interesting as well. I think.
Question for Nina, if that's okay.
Yeah.
So really interesting kind of teaser that you gave there. I was just wondering why you kind of started looking at ATG5 to begin with, and whether you kind of have a working hypothesis now moving forward for why it might be inhibiting eosinophil effector function, because certainly if it's doing that, it could be, potentially a really interesting therapeutic target for future studies.
Yes, hello. Thank you for the question. So why ATG5? To answer first, it's because the lab works on autophagy and was already looking in the ATG5 previously, also in neutrophils. And indeed, it was very interesting to see the differential effects also of ATG5 knockout between neutrophils and eosinophils, seeing that, for example, knockout in neutrophils accelerated and caused accumulation in the different organs, while knockout in eosinophils caused delayed and reduced differentiation. And it was also interesting as well, for example, for neutrophils and mast cells, reduction of ATG5 or ATG7 also reduced effector functions, while in eosinophils, it was also to our surprise that it enhanced, for example, degranulation activity as well as bacterial killing. We don't really have the exact mechanism behind. We also looked for the activation of some pathways, for example, p-STAT3 as well as p38 and p44. We saw that following the priming with, for example, GM-CSF or IL-5 cytokines, we could see increased activity of these pathways when we checked that by Western blot. So we thought that might give a hint for the increased activity of knockout in this case.
Really interesting. Thank you.
Thank you for the question.
Any other questions? How about I found that Josh Mitchell had put I think a small eosinophil figure. I think it's a light microscope image that you had put. It seems that the morphology is different as well with IL-5 and IL-33 trigger. Can you just shed a little light on that? Was it a light microscopy image?
Yeah, the morphology does change quite dramatically too. So from kind of our peripheral purified eosinophils, they're nice and spherical. And then when you do acute treatment with IL-5, you see a very distinct polarization where the nucleus moves to the back of the cell, kind of forming this nucleopod. And the granules are at this migratory front where they make up this main portion. And it takes on this kind of acorn-like shape. With IL-33, with this acute activation, what you see is ultimately there's kind of almost this initial activation state where, unlike perhaps the more distinct, sharper features of IL-5, it's a little bit more oval shaped. And then eventually, as it becomes more adhesive and more migratory, it actually flattens out into this larger pancake-like cell, which forms this very thin layer, probably about 1 or 2 microns in thickness. And it's this monolayer of granules. And the migration is also much slower than the rest. So that's been one of the inherent interesting questions for us and trying to tie in some of these features. You know, I know it was a 1-minute tease to practically 9,500 phosphosites. So it's a
great. Okay, now that makes sense. My next question is to Timothy, unless anybody else has a question.
Oh, sorry. Go ahead, Julia. I had a really quick follow-up question for Josh, which I'm just curious to know where you're obtaining your eosinophil culture. Is it differentiated from bone marrow progenitors or?
Oh, blood? Okay, yeah. So our eosinophils, right? So these are all patient purified eosinophils.
Okay.
But what about the fact that they may already have had the effect of IL-5 or IL-33 in their system, whether they're steroid naive, whether atopic, non-atopic, or healthy?
Yes. So the patients that we're getting our eosinophils from usually have some sort of allergic condition, which just gives us the ability to actually purify them, right? Outside of that, we do have on a patient-by-patient basis, right, kind of a lot of that health information. Oftentimes, at least from what we found, right, when we get our eosinophils, they are kind of the spherical unactivated state. You know, I can't say I've looked very heavily at each individual state. I will say, at least for the sake of kind of the experiments that we found, is that we've had very good consistency, not only with the morphologies, but also kind of the seemingly activated type between them, right? We always get they're allowed to rest too, which I guess can in some ways help. But we've generally found that from the peripheral blood, we've had very consistent, at least morphological presentations.
Have you looked at least looking at the activation markers like CD63 to make sure that they are unactivated, considering you're getting it from patients? Yeah, so we do. There's some immunofluorescence we look at. I can't say that we've strictly done flow, looking at all of the surface markers before all these. So that's something that one of the things I do want to look at is take a comparison across kind of this wide variety of patient samples, take a look at them. Thank you, Josh. We have about 3 minutes in this breakout room before we have to go back to the main session to wrap up today's webinar. So in the last 3 minutes, if anybody has any questions to any of the speakers or any of the 3-minute abstract speakers.
I have a question for Josh, but it's not actually about his abstract. I've been thinking a lot about eosinophil nuclear shape. And so I guess I was wondering, in any of your kind of studies, so you mentioned about the nucleus kind of getting pushed to one side of the cell in certain activation conditions. I'm wondering, have you seen conditions where the shape, dramatically changes? Maybe like recircularizing or becoming more fragmented, or any kind of phenotypes to that effect?
Oh, that's a great question. So, I mean, in some ways, so I don't see any changes per se in segmentation, right? Usually the two lobes do seem pretty consistent across the activation state. I will say, in respect to what cytoskeleton, how that might be putting pressure on the nucleus seems to be kind of interesting, because with IL-5, right, I mean, it really jams in. So I don't have the images up, of course, but in confocal microscopy, right, when you look at DNA staining, really jams into the back. Those two lobes are really squeezed together. And IL-33, the nucleus flattens out more, right? I guess it has more room to spread. So at least in that respect, you do see perhaps the compactness or the maintenance of the nucleus into a tighter positioning. That certainly changes between those two states.
Interesting. Thank you.
Okay, we have one last question. Anybody? We have to leave the breakout room in about 120 seconds. So any last question for anybody, any of the speakers? I can see Sina Karimi has also joined the call, who had a 3-minute abstract. So any questions to Sina, who just worked on the mepolizumab efficacy of the T-cell driven HES? So if that is it, then I'm going to close the floor for now. Please feel free to leave the breakout room and go into the main session where Dr. Florence Rufos will be taking the session. Thank you.
In the meantime, I'd like to welcome you back to the main session. We only have a few 10 minutes, 12 minutes left. I hope you enjoyed the presentations. I hope you were able to discuss the different abstracts as you would have liked to. So there are 44 of us now, so I think we can continue discussing a few points. So I would ask at this stage, perhaps Kay or Andrea to start presenting the slides. Perfect, thank you. So closing remarks, besides welcoming you back, I would also just like to ask you to be careful to incoming emails these next few days. We will be asking for feedback on the session. This was our very, very first abstract session. We don't know how epidemics are going to evolve in the following months and years, so we may have to do this again. So we'd be very, very pleased and grateful if you could send us feedback on the way this abstract session was held. Overall, I think attendance was successful, but we may have had a few frustrating aspects for certain people. So if you could share the way that you would have liked this abstract session to happen, please respond to our little survey afterwards. I'd now like to introduce you to our two recently nominated trainee representatives. So this is on the next slide. You will see their names. We have Kiho Sun on one hand from McMaster University, who's one of the representatives. And we also have Eva Knupresch, who is currently working at the Medical University in Graz in Austria. They are both postdoctoral fellows. They're both working obviously on eosinophils and eosinophil biology. And I would really encourage you to pay attention to emails that you may be receiving from these two people as they may be proposing different activities for trainees. I will now leave you with Eva, who is going to start her very first mission as trainee representative, and she's going to announce the award winners of today's abstract session.
Thank you, Florence. So hi everyone, I'm Eva, and together with Hiho, we serve as training representatives, as was mentioned. I think I speak for both of us when I say that I was amazed by the quality of today's talks and presentations, and I think that the future of eosinophil research looks very bright. I was given the task of announcing today's winners. These winning presenters will be awarded free registration for the next on-site IES meeting, as well as they will receive a certificate via email. I have to point out that our reviewers really had a difficult job choosing the top scored abstracts. So without further ado, your winners are listed on the slides already. First winner is Tetsuo Shoda for his work on establishing eosinophilic colitis as a separate disease entity, which is distinct from other EGITs and IBS. Next up is Natalie Ben-Baruch Morgenstern, presenting single-cell RNA sequencing of mast cells in eosinophil esophagitis. Then we have Julia Tenenbaum for her research in FIP1LA1 PDF GFRA clonal HES, which is responsible to imatinib treatment, and Marijuan Hassani for presenting the effect of mepolizumab on eosinophil kinetics in patients with eosinophilic asthma. And finally, last but not least, we have Sina Karimi, who presented mepolizumab efficacy and impact in patients with T-cell driven hypereosinophilic syndrome. So big congratulations to all the winners. I look forward to meeting you as well as hopefully other trainees at our next live-in meeting. Thank you.
Okay, so we're approaching the very end of this abstract session. I would just like to remind you of our logo, email, website for the International Eosinophil Society. And as you can see on this slide, our society's mission statement shows that education and exchange pertaining to eosinophil biology and eosinophilic disorders are very key and essential for us as a society. And we hope that you agree that today's abstract session has actually met this goal. We will continue to hold our monthly webinars as shown on the next slide. I think there was another slide somewhere between the two, showing the titles and the dates of our next webinars. I don't know, Kate or Andrea, if you can show that slide. So the next webinar will be held on October 27th. It's dedicated to eosinophil functions and diseases. And we will also have webinars in November and December. You can already see the title of the November session here. Please don't forget to register. Registration is free, but it's important to register to be able to have the link and to be able to interact during these sessions. I would like to end with a special thanks to those that have helped make this abstract session possible, and especially to Paris Khoury and Manali Mukherjee. Their ideas and their enthusiasm have been absolutely great and have made this possible. And I think it was a very successful meeting, thanks to them. We'd also like to thank Seema Sivis for moderating the Gastrointestinal Disorders Room. And we would like to thank Andrea and Kate from EDI for managing all the logistics. This was a tricky one. There was more complicated logistics than usual for this abstract session. We thank you very much for being able to even react while the meeting was actually being held to a few technical glitches that we had. So thanks to everyone, thanks to speakers, thanks to the abstract presenters, and I hope to see you again in a little less than a month.